Alright, welcome back. I'm going to do a quick recap of section 3.1, which was about the cell cycle. So, the blank is the regular pattern of growth, development, and division in cells. The cell cycle. Identical strands of a chromosome are called sister chromatids. Two identical nuclei are formed during mitosis. I guess you could also say mitotic phase. Cytoplasm divides to form two cells during cytoplasm. Two cells produced during the cell cycle are called daughter cells. The two main phases, in the cell cycle are interphase and the mitotic phase. After the chromatin is duplicated, sister chromatids are connected by a centromere. The most important result of the cell cycle is two daughter cells, or identical cells. The cell cycle is important in, living organisms, multicellular organisms. The cell cycle is important for growth and repair in, that would be multicellular organisms. After division, the parent cell it no longer exists. Okay, it has become two daughter cells. List the three stages of interphase in order: G1, S, G2. On another slide, what stage of interphase is the period of growth and development? That would be G1. Which stage of interphase is where DNA is replicated? That would be S. Which stage of interphase does the cell prepare for cell division? G2. During which phase do the nucleus and cytoplasm divide? That would be the mitotic phase. Ooh, lots of review there. All right, so today I'm going to focus in on the phases of mitosis. So we are in the mitotic phase. I'm going to focus on mitosis. This is the division of the nucleus. It is broken down into 4 steps, okay, or 4 phases, if you will. So the first phase is called prophase, and our copied chromosomes that were spaghetti-like before have condensed and formed these X-shaped chromosomes, okay? So two sister chromatids held together by a centromere. So you can actually see these duplicated chromosomes in a microscope. Some other things that happen in a prophase, in prophase, is that the nuclear envelope or nuclear membrane that makes up the nucleus starts to break down. So you can kind of see that they are making it kind of dash-like. It's going to break down. And then we have these organelles called centrioles that form these spindle fibers. And they're going to form. And what these spindle fibers are going to do is they're going to go to opposite ends of the poles. And then with their spindle fibers attach to the chromosomes and maneuver the chromosomes so they can separate them equally. So this is a cartoon diagram of what prophase looks like. And when you look at it underneath the microscope, you can see that they're condensed. You can see blebs of them, concentrated areas of these condensed chromosomes. After prophase, we have metaphase. And this is where our spindle fibers have attached to the chromosomes, and they're going to maneuver them and line them up down the middle of the cell. This is the shortest phase of mitosis. So metaphase, middle. That's how I remember metaphase. Chromosomes are in the middle of the cell. So this is an animal cell. Okay. That's the cell membrane. You can see we don't see a nucleus. You can kind of see that we have these fibers radiating out of this area here, and then fibers radiating out of this area here. So those are the spindle fibers. And stained in purple are the chromosomes lined up in the center of this cell. After metaphase, we have anaphase. And so one way I remember anaphase is away. The sister chromatids that were held together by the centromere that you see in metaphase, they're going to be pulled apart from each other. Okay, so away, apart, however you want to describe it. So two sister chromatids in each chromosome, they separate from each other. And once they're separated, now we just have two identical single-stranded chromosomes. So here's a plant cell, because it's boxy. And you can see that we have our chromosomes that are being pulled away from each other. They are no longer duplicated chromosomes. The sister chromatids have been pulled apart. After anaphase, we have telophase, and this is the final step of mitosis. Our spindle fibers are going to disappear. For example, back to the centrioles. Our chromosomes are going to go from X's or single-stranded to spaghetti again. And then another thing, 2 new nuclear membranes form around the chromosomes. So telophase, I think of 2 new nuclear membranes form. So here we have an animal cell again, and there's our chromosomes are going to start to uncoil and become spaghetti-like again, and it's just about ready to pinch into 2, which leads us to the second stage of the mitotic phase, which is cytokinesis. The cytoplasm divides into 2 cells. Now, one thing you really need to know about cytokinesis in animals and plants is that it's different. Animal cells don't have a cell wall, so they can literally pinch into 2, okay, and pinch all the way through so that it touches the other side, and then it just like pinches, I don't know. Just think of it as a belt. And you take this belt and you wrap it around this cell, and you tighten the belt, tighten the belt, tighten the belt, tighten the belt, and eventually, pop, you get 2 daughter cells. Okay, so we call this structure a cleavage furrow. Plants, well, they have that cell wall. You can't stick that belt around it and tighten it and pinch it into 2 because that cell wall is so rigid. So how does the plant divide into 2? It forms a structure called a cell plate, which is formed by a bunch of vesicles, and they will fuse together and then meet up with the cell wall, as you can see here. So a bunch of vesicles forming the cell plate, fuse together, join up with the cell wall, and bam, 2 new cells, 2 new daughter cells. Okay. Oh, my Mac's gonna die here. So the results and reasoning behind cell division. So in mitosis, at the end of it, you end up with 2 genetically identical cells. Human cells have 46 chromosomes. And when it divides, it produces 2 cells with 46 chromosomes. So it helps to make sure that, DNA is correct. It's important for reproduction, growth, and replacement and repair in organisms. Reproduction, especially in unicellular organisms, growth, replacement, and repair in multicellular organisms. So, there I go again. So for the reproduction, that is for the unicellular organisms. So what I mean by unicellular, they're made of 1 cell. They're usually prokaryotes, like bacteria cells, E. coli, germ cells. Growth, replacement, and repair are very important in multicellular organisms. So growth, it allows multicellular organisms to grow and develop from that 1 cell. We have replacement because you lose cells all the time. Your skin exfoliates skin cells. They become wore out and damaged. Your stomach cells replace themselves like almost every 3 days. Repair, when you get a cut, a paper cut, a bruise, there's damage done to your body, but not all that damage can be, repaired, and that's what you get with scars. Okay, so that's 3.2 notes.